Assalamualaikum. Boleh dengar saya ke?
Waalaikumsalam, boleh?
Boleh eh? Are we expecting more to come in? Ke ada yang tak dapat join pagi ni? Eva?
Tak ada yang sakit setakat ni, tak ada yang baik lah.
Okay. So nak tanya, shall we proceed or masih nak tunggu ke?
Proceed je kot tu, Prof.
Okay. Yalah, because this is out of our normal punya slot kan ya. So just in case some of you ada hal ke. But whatever it is, if you all can fill in attendance. Laisi kan? Ada kan? Did I pass you attendance? Ada ya? Untuk hari ni dan juga esok lah ya. So I hope you do not mind me not opening my camera sebab berat pula anak I semua kat rumah ni. 88 orang sekali, my husband semua guna internet. So it's quite unstable. So hopefully you don't mind it. Boleh dengar saya clearly kan?
Boleh.
Okay, alright, okay. So Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Let's begin our session with recitation of Ummul Kitab. Okay, so Alhamdulillah, we meet again. So apologies for not being able to do class in the normal slot. But I think we are on our second last session. Tomorrow, satu lagi lah sekejap saja. So hopefully everything is in order. And then anything to do with the perjalanan of this course, everything is alright. Anything to update me, Eva? Ada tak anything to do with toxicology yang belum selesai ke with the other instructors ke? Macam mana? Semua dah settle?
Semua dah settle kecuali yang writing up tu.
Yang assignment saya lah ke macam mana?
Yang Prof.
Yang saya punya So that has a deadline of tomorrow kalau tak salah saya.
Yes.
Betul ya? So yang lain-lain hopefully you all have settled lah ya, macam report semua, lab ke, tak ada masalah ya?
Tak ada masalah insyaAllah.
Okay. But the last time I check, you punya camp dalam academic system tu, masih ada saya punya je markahnya. But some of the instructors have mentioned that they are working on marking your assessment ke apa ke. So nanti they will fill in masukkan markah So hopefully you are able to actually contact the individual instructors to know the progress lah. So mungkin Eva boleh bertanya juga dengan yang the other lecturers.
Okay, boleh.
Okay, alright. So meaning for me, kalau tak salah saya, yang mid-sem pun dah masuk dalam camp tu. And then of course I will assess your write-up lah yang you akan submit tomorrow. So hopefully you will have no problems with the turn it in ke or submission ke. So hopefully it's clear lah in the Padlet, the instructions. Okay, so anything just contact me lah. So I'm going to share my screen macam biasa. So hang on Okay, boleh nampak ke? Brothers, sisters? Can you see?
Yes.
Yes.
Yeah, okay. So I think we are relying on this Padlet wall. This will be the last Padlet wall pun, that I am sharing. Yeah. So just now tak payahlah I buka teaching plan ni. So masalah write up ni tak ada. So you are able to actually read the paper, which I've attached here. And then last time we do in vivo kan, kalau tak salah. Buat in vivo kan? Betul ke? Remind me. Macam dah lama tak jumpa you. Am I correct? We did in vivo, yeah? So this time we do in vitro lah. Betul brothers, sisters, yang hari tu kita talk about in vivo so that I don't repeat myself lah. Can you hear me? Ke internet saya tak stable? Can anyone respond? Boleh dengar saya ke?
Boleh.
Boleh okay. So the last time memang kita intern you are saying yes, the last time we did in vivo lah ya. Okay, so macam ni, before I go further, okay. Mana nak tutup ni kejap okay, so kalau just in case okay, sorry, boleh dengar saya ke? Aduh. Yeah, okay. Hang on. Okay, can you see my screen now? Brothers, sisters, boleh nampak
Yes.
Okay, alright, very good. Okay, sorry lah, macam ni lah kalau relying on technology, But I think I straight away go into the main points lah. I do not know whether you have actually looked at these sets of slides, but the last time we talked about in vivo, and then you are doing a write-up, especially looking at whether or not in vivo tests are necessary, still necessary, But today we will focus on in vitro lah, so I will want for you to actually look at this website perhaps, so If you are looking at my screen, ini terlampau kecil ke? Okay ni. Should I besarkan sedikit?
Okay je.
Okay, right? Okay. So I think we have talked about the 3R principles lah previously kan, yeah. So how we are trying to be humane in our experiments, especially for us kan dalam biomedical science department ni kan, biomedical science program. So how we conduct our experiments, our research, yeah. So hopefully we are responsible. We are responsible about it. Yeah. So that is why we are always thinking about how to actually be humane to reduce perhaps the number of animals that we are using, yeah, in our research, to actually look at our our methodology, how we can refine it to be better. And then kalau let's say we are implicating pain or causing suffering of our animal subjects, how can we actually improve our methods, yeah, so that comes under refinement. And then of course how instead of using living organisms like the animals, yeah, perhaps we can even use like computer simulation ke, modeling ke, or even use cell culture, so that comes under replacement. So yang ini we have talked about this kan before ke belum lagi? Students, about alternatives ni? Ada tak kita bercakap dalam pharmacology ke dulu?
Rasa macam belum, doktor. Tak pasti lah.
Rasa macam belum ya. Okay,
Belum.
so maknanya okay, so I open the chat box there so that I can see ya, kalau ada anyone nak straight away apa ni tanya questions ke, ya. So okay, so like what I said just now, meaning we must look at our approach and whether or not there can be alternatives, yeah. So we must be humane, yeah, in what we practice in our experiments in our profession as biomedical scientists, yeah. So when dealing with animals, yeah, especially in our experiments, yeah. So the 3R principles becomes very important, yeah. So it's basically reduction, refinement, and replacement, yeah. So ini memang prinsip-prinsip yang you all kena guna lah, ya. So nanti let's say in year 4 kan, or perhaps year 3 you dah start buat your research project, ya. So maybe some of you will be using animals, yeah. So we'll be using organisms, yeah, living animals, yeah. So you perhaps will need to send your research proposal to the Animal Ethics Committee of Kuantan campus, let's say, yeah. So they will be looking at this, yeah, whether or not or perhaps kalau guna animal species apa you guna, and then berapa banyak, ya. So all this will be assessed by the Animal Ethics Committee for Kuantan campus, yeah. And then they will look at perhaps are you applying this 3R principles, yeah. So we always go for this lah, yeah, a reduced number of animals, yeah. So seboleh-bolehnya, yeah. So maknanya kalau you use your research ni has a certain goal. Let's say you are testing an anti-cancer drug, let's say kan, contoh, okay. So you can study what sort of species of animals that you need to use. Yeah? So are you familiar with the mice bred in lab? Perhaps are you aware that that there are a lot of species that researchers have used in experiments?
Yes.
Ada kan ya? Macam ada tikus putih, ada tikus hitam, kan ada yang tikus yang macam bugil tu, yang newt mice, ya. All these animals are bred in the lab, for the purpose of experiments lah, macam let's say tadi I bercakap pasal to test anti-cancer drugs kan, so you really have to like look through the available species for experimental punya purposes, perhaps you can use the nude mice, so to inject cell cancer kat belakang dia ke, and then test the anti-cancer drug, so maknanya you have to study the animal subjects that you are going to use in your experiments, so seboleh-bolehnya you memang kena identify which species to use and how many lah, so semua ni later you akan present to the Animal Ethics Committee for their assessment lah, because sekarang animal rights ni makin kuat. Dulu while I was doing my studies in the UK, so the animal rights group are very strong there. So they will actually picket in front of the universities, kan, and then they can even throw stones at researchers. So I was doing my studies in Bradford, UK, so memang dekat UK memang animal rights punya societies ni very strong, so macam saya pun I nak kena tolak my trolley full of mice tu naik tangga belakang sorok-sorok, that sort of thing, but I think you can understand lah why animal ethics is also equally important as like macam human ethics kan, human research ethics kan, because memang animals ni it's a living thing kan, so but Allah dah jadikan animals tu for us to use, there is a purpose there, so think about this, so whatever you do later, try to be humane lah, so do research but do responsible research, okay, so coming back to the slides that you can see, refinement ni tadi I bercakap pasal the methodology, so you really have to look at your experiment methodology, so look at whether or not your procedures tu akan cause any pain ke or suffering to the animals that you are using, so you have to employ things like using anesthesia ke, so to make it to reduce the suffering of the animals, so those are the important points under the refinement punya principle lah, as for the last principle, it's replacement. So just now I did mention perhaps you can use computer modeling, simulation ke, and then what else? Other things lah, macam cell culture. So I think I have mentioned juga to a certain group of students. I give an example, macam dulu I wanted to do an experiment on keratocytes. Keratocytes tahu ke? Brothers, sisters, keratocytes tu apa agaknya? You can even search right now. Sekejap ya, I tutup tingkap ya. People are doing work outside. Sekejap okay. Okay. Keratocytes. Apa tu neuron ain? Keratocytes. Keratocytes kan? Cornea cells kan, cornea cells kan, so macam ni, my team wanted to do an experiment on the cornea cells kan, cornea yang mata. So we selected rabbit punya cornea cells, ya. So rabbit ni kan cute kan, so we were quite reluctant to actually macam beli a number or group of rabbits and then just to do research on cornea punya cells. Yeah? So what we did after that is to actually pergi ke restoran, restoran yang jual daging arnab. So we get the the chef to just give us the head of the arnab yang diorang baru sembelih lah. And then we took the cornea cells and we culture, and then we test whatever we want to do on the cornea cell culture. So do you see? Can you see how we apply this this 3R principle? Yeah, boleh faham
Yes.
Yes, so it would be much better than, let's say, you beli arnab lab ni, dah lah mahal satu pun sebab arnab yang or animals yang bred in the lab they are expensive lah so and then kita just guna cornea cell mata, yang lain buang macam tu. So it's very wasteful. So you have to think of ways how you can actually be responsible about doing research and you can apply this 3R principles. Boleh okay, so itu memang satu contoh which I think is very good lah. So and of course kita tak guna banyak arnab hari tu, and just kiranya we test on the culture, the cell culture. So it's a very good example. I hope you can understand lah, Okay, so when we go on to look at the use of animals in the lab, this has to do with policies, law, regulations. So I think you can read about this lah. So there are a lot of macam committees or legislation punya committees yang look at or give out approval or permission or how to conduct especially animal studies lah. So kalau any of you ada any questions, you can just simply put in the chat box so or perhaps you can interrupt me lah, open your mic So meaning this if you employ the alternatives that we talked about just now so meaning you are doing good planning of your research so meaning there's no wasting so we go for this lah, you don't waste, and then you do pilot studies. Are you all familiar with what it means by pilot study? Pernah dengar ke? Anyone? Pilot study ni kan macam we test with a small group of animals dulu ke, so we do screening so that kita tak membazir so that is why it falls under the reduction punya alternatives lah so that's equally important and it shows or it indicates good planning of your research or studies. So just now I cakap pasal strains of mice ke species ke, so you really have to study so jangan sampai tersalah guna species yang let's say tak sesuai dengan the objective of your research. Okay, so that is something that you have to think about lah. So you really have to research on the animals that you are going to use in your study. So yang refinement memang I think by now you understand you have to look at the methods whether or not you are going to cause pain or distress to the animals So dulu I sampai pergi ke Singapore belajar pasal husbandry, animal husbandry. You find out what that means Animal husbandry is simply the care of animals that we use in our experiments lah especially in the biomedical science fields So sebenarnya we really have to take care of our animals that we use in our research. Kena macam air makan tu biasalah, wajib semua, but tempat tinggal dia, and then sama ada we provide macam setengah-setengah animals ni perlu toys ke, so that sort of thing, so I even went to Singapore to learn about those things, so they do a massive training for people doing animal studies, so it's very important that housing conditions in the for the animals are looked after, because you know why? Kalau let's say you tak jaga you punya research animals, and then let's say dalam satu box tu ada dalam satu cage tu ada like a few mice, kalau you tak bagi makan ke, they will eat each other, let's say, so it happens, because the last time we even have postgraduate students who are not responsible, and we found out in the cages that one animal eat another animal, so cage tak bersih, so that sort of thing, so that's why you when you conduct research, you have to conduct it responsibility and be accountable. Okay, so the last this one is the replacement. This is replacement lah, so ini yang I bercakap pasal using other methods, using even in vitro methods, to actually replace as an alternative of replacement. Okay, so boleh juga lah guna macam the lower organisms like even microbes ke, so think about what you want to do in your research, so sebenarnya the planning of your research will take a long time, when you have everything in place, then it's easy sebab you can straight away do your research and then collect data and analyze. Okay, so that would be my advice lah. Alright, so when we talk about in vitro methods, sebenarnya dia bukan like a total replacement lah, of in vivo methods. I think this you have addressed, if I'm not mistaken, in your assignment, so you can read about this lah. So it really depends lah on the objective of your study. I think you understand now when you set out to do a research, you really have to think about the objectives of your research, okay? Because the objectives of your research will determine what sort of methods you use. So kat situ ada kaitan dia, so maknanya you kena set up nanti when you do your research project, you have like one or two objectives, so setiap objectives tu akan ada its own methodology, procedures ke, so the results from the methods, the experiments that you do later will tell you whether or not your objectives have been achieved. Okay, boleh dapat ke tu? Boleh, brothers, sisters? You can follow,
Yes.
Itu points yang sangat penting. Mungkin you can carry on until I think you need to remember what I said when you are embarking on your research project in your later years later, Okay, so think about that, Alright, so while we are talking about replacement still, ada banyak-banyak jenis ni replacement, so nanti you can have a look through lah, there are things like absolute replacement, relative replacement, macam yang saya tadi, yang penggunaan RNAP tadi, yang ni kan, what do you all think? Perhaps it comes under relative replacement lah, sacrifice one rabbit, so that it can provide us with cells or tissues for in vitro studies. So what I was doing perhaps can come under relative replacement lah, but there are things like partial replacement also, okay, so I think you can read about that, but there are also alternatives like direct replacement, so instead of using guinea pigs, in vivo, there are cultured human skins, that can be used in the lab, in vitro, to test, perhaps test substance, but there are also other types of tests which comes under indirect replacement, So instead of using rabbits, and then test for paragenic effects, so use another species ni, Limulus amibusai ni daripada belangkas, to actually give you the same output. Okay, so we'll talk later about that lah. Pernah tengok ke belangkas ni?
Pernah.
Pernah ya? Kita boleh makan ke belangkas ni? Brothers, sisters.
Ada je orang makan.
Mungkin telur
Boleh.
lah, telur dia. Ya? Tapi dia ada macam apa ni, macam dia punya
Poison.
these are like dangerous animals also. Dia ada macam apa ni kat depan ni, so these are horseshoe crab. Kenapa crab pula sini? Crab, Okay, so sebenarnya dalam this species ada a substance, that can help us in terms of identifying paragenic punya effects. So I think you can read further about Limulus amibuside lysate test. So this is in the horseshoe crab. So there are amibosides yang ada granules, yang ada clotting factor. So they are in the blood of the horseshoe crab. So memang kiranya kita ambil this from the crabs, and the crabs are returned to water lah, and then kita gunalah that substance to actually test for effects of any other agents, so dekat situlah inovasinya, innovation. So I think it's quite a good advancement in biomedical science, from a long time ago. So perhaps some of you will also do like cytotoxicity test later in your research project, using cell culture. So yang ini nak identify lah, dead cells ke, alive cells ke, cells which are apoptotic, okay. So using the MTT assay, so it uses macam color as indicators, so that's why it's a colorimetric assay. So using dyes semua tu. So in the end you can actually come up with a percentage banyak cells mati ke over those who are still which are still alive, so these will be like the data that you will collect later lah, Okay, so itu macam contoh-contoh, so perhaps you can read around the area ke, or find out lah, read more about the test methods just now, okay. So other types of replacement alternatives are also being used, in terms of to test safety of chemicals. And every day we are manufacturing a lot of chemicals for a lot of or perhaps a variety of uses, but since the consumers are usually us humans, so it's very important now that we do safety and toxicity testing. Remember from previous times, pasal safety, pasal toxicity ni, zaman dulu it's not very strict, but itulah macam yang let's say some medications ke apa ke given to pregnant mothers, and then they all deliver babies without hands and feet, so remember the thalidomide punya case, so it's one good example whereby all these substances or agents or chemicals which are used by humans, they all have to be or perhaps undergo safety and toxicity testing. Okay, so macam sekarang, what is hot topic is obviously the vaccines, Are you well versed with the COVID vaccine, the progress, the types? Brothers, sisters, baca ke tidak?
Baca.
Baca ya? Okay. So be aware because it's very much related to us in the biomedical science fields. Yeah. So I think by right you should know what sort of vaccines that we have around now for COVID-19 kan ya. Kan ada banyak-banyak jenis ya. So they are of various types and formulation. Okay. So how quality is being controlled because it will actually reflect on whether or not they'll be effective against the virus. Yeah. That is why it's important to do testing. Yeah. Okay. So that is also very much related to what biomedical science fields are. Okay. So let me introduce to you also other type of testing. Macam ni kan penting pada kita. We need to test whether or not substance yang manufactured by us can cause skin corrosion ke. So zaman dulu we test on animals macam ni lah. Yang rabbit semua ni. But now, of course, this is something that we look upon with horror, kan ya? Lagipun kesian. That's why we changed to in vitro. There are like artificial skin cultures that we can test chemicals or substances on. So that will be much more humane lah, ya. And then zaman dulu juga, we always test macam effects in vivo. So kesian lah ya. So now we have alternatives. So like the membranes, chorion, allantoic membranes to test. So it's much more humane lah. Then of course instead of using like mice, perut dia kita busungkan dia. Okay. So we can do in vitro. So all these are replacements which are very much humane lah. Okay. So this is production of monoclonal antibodies for a lot of other testing. Okay. So see the ones that we talked about just now. So read around the subject matter and then making use of hormones, vitamins, to actually give us the same results for the same objectives. Okay. So boleh faham ya? So if you need to ask me questions or share anything, you just simply do. Okay, so let us look at advantages of in vitro tests. I think a lot of this you understand. You know, or perhaps you know that these are like pros of doing in vitro tests. So kalau kita buat in vitro, definitely we can control the testing conditions. And kita tak ada macam difficulties or perhaps disturbance by systemic effects because when we do a whole animal punya study dalam in vivo kan ada banyak faktor dalam badan animal tu yang akan affect your results kan but if we do in vitro kita boleh control the environment and we don't get a lot of systemic punya disturbance lah. Boleh faham ke systemic ni apa? Brothers, sisters, do you understand me? Systemic. Macam one whole body kan, systemic so make sure you understand, okay? So, so kalau we are able to control the testing conditions, so definitely we are able to actually maintain the factors, so we reduce any sort of variability lah in between experiments. But the point number 4 ni, why I put bracket dalam bracket ni cheap ni, sebab kadang-kadang kalau kita guna test kits pun in the lab, they are not cheap so macam hari tu saya nak test DNA fragmentation kan. So even to test like 50 samples, that was like 10, 12 years ago. Satu kit yang supplier jual tu at that point in time is like 3,000, 2005. Untuk test 50 samples saja. So can you imagine? So it's not really very cheap lah. but testing can be fast, yeah. And we can get results fast when we do in vitro punya test, yeah. So that is something that you can talk about now, so yeah. And then no need big amount, yeah. And then of course, since it's not a systemic, a whole animal punya procedure or study, so we can perhaps limit the amount, or perhaps we get a not so much amount of waste, yeah. So these are like what can be used in vitro. Okay, so obviously we reduce the testing in animals. Okay, but obviously, yes, ada nak tanya question kan? You can chat, okay, so that I can address, yeah. Okay. So tadi yang pros, but obviously testing in vitro ni ada cons there, yeah. There are limitations, okay. So I think you can go through this lah, yeah. So okay, so macam in vivo dose response semua tu, it cannot be obtained, yeah. You cannot evaluate lah, of course, yeah, the systemic whole animal effects ke, yeah. And there is like no interaction between tissues and organs, yeah. And remember what we learned about in pharmacology, pharmacokinetics, yeah? So that is also out of the picture, yeah, and other things lah, okay. And of course, macam kalau kita buat in vitro, let's say we do cell culture kan, sometimes we are not able to like passage the cells, grow a new one ke sampai berpanjangan, cannot, yeah. So usually chronic effects cannot be tested lah, yeah. So that is like the downside of doing in vitro, yeah. Ada apa-apa ke nak tanya saya at this point? You're alright, Okay. Kalau boleh you off your mic, so that it doesn't interfere with the class. So you can chat first, Okay, so I move on to look at other replacement alternatives to test, for instance, genotoxicity, yeah. And other things like the tolerance, yeah. Macam tadi we looked at skin corrosivity, yeah, eye irritation, yeah. But some of the tests memang we can do macam screening, toxico screening, yeah, and also absorption through the skin, yeah. So there are a lot of replacement alternatives, yeah. But certain things kita tak ada replacement alternatives. Macam mana kita nak buat pharmacokinetic study kalau guna in vitro, yeah. So this has to be in vivo. And if your objective of your research is systemic toxicity, how can we do it in vitro? Yeah, so that also needs in vivo. Yeah, so I think you can understand this, yeah, as I go through this later. So of course, whatever protocol or experimental approach that you use, yeah, so hopefully it's a validated type of method, yeah. So dulu I ada bercakap pasal OECD, yeah, so maybe you have had time to actually look up also, yeah. So these are like established and validated tests which you can use in your research. So maknanya dah establish the protocol, dia punya methodology, yeah. So you can look up, and if your objectives is applicable, so you can even just simply straight away use the approach, yeah. So this one I think you can look up also under OECD pun ada, yeah. So that is under chemical testing. Biological testing is here, yeah. Genotoxicity testing, all these are like established experimental protocols, okay. All right, so this is also one which is very interesting, yeah, to look at cytotoxicity, yeah, of chemicals, okay? So we can use in vitro, yeah, using also I think light, okay. All right, so I have put it here for you to be able to read later, yeah. And then just now I talked about this also, artificial human skin, which you can use to test chemicals. But here you can see it's being tested for cosmetics. Yeah, so very useful. Yeah. Okay, so then we move on to yang ini pun tadi ada ya dalam the list ya, Toxicity. Yeah. So I think this becomes very important to us lah ya. You are aware of chemicals or substance. When we are exposed to them, they can change our DNA. Yeah, so they can also be not only mutagenic but carcinogenic. Yeah, so this I think is a concern to us lah. Yeah, because I understand also that you all students, you are aware kan, macam the effects tu kita tak nampak immediately. Macam let's say some people they get cancer kan ya, but from what? From exposure to what? Yeah, and we don't know sometimes. Yeah, and when was he or she exposed to that carcinogenic substance? We don't know also, because most likely the effects from the exposure is long term. Dah 10 tahun baru manifest. Yeah, so I think boleh fahamkan tu, brothers, sisters?
Boleh.
Boleh ya? Okay. So
Boleh,
those
Prof.
are the things that you should think about. Yeah. But I have given you some definitions here, which I think you can refer. Okay, thank you. So you go through lah ya. So really look at the definition of mutagenicity. Apa maksud dengan genotoxicity? Yeah. So let's see here, mutagenicity, referring to permanent changes in the genes, yeah, in your genetic material, yeah, and dia boleh be inherited by the younger generations macam tu, yeah. But certain definitions is much broader. Macam here, genotoxicity memang akan include sekali mutagenicity, yeah. So what is it is all about, you can have a look here lah. But there are also the relationship between genotoxicity and carcinogenicity that you have to think about, yeah? Because sometimes carcinogens are non-genotoxic, yeah? So some substances are genotoxic but they don't cause cancer. So itu yang masuk second point here, yeah? But some substances or chemicals or whatever that we're exposed to, they are both genotoxic and also carcinogenic. Okay, so itu yang kita nak kena fikirkan ya. And as biomedical scientists, perhaps it's important for us to be doing the testing as well. Okay, so for the last group there, yeah, so they are positive in genotoxicity tests, yeah, and positive also for carcinogenic studies, yeah. So that makes them the most dangerous of all, yeah. Okay. So there are tests which you can use, yeah. So I'm sure you have heard of AHBS test lah. Ada tak in any of your other courses kan? You look at this. Tak ada. Brothers, sisters, pernah dengar ke any of the standard tests? You can even type your answer. Yeah? Tak pernah ya? Okay. Yalah, lagipun kan sekarang dah lah macam 2 tahun you all tak datang ke campus buat lab semua, so it's especially difficult, yeah. But the knowledge is out there. It only takes for you to actually click on YouTube, look at AHBS test, what is it all about, yeah? Or perhaps look at mouse lymphoma assay test, yeah. So contoh yang kalau AHBS test ni memang digunakan to look at gene mutation, and kita guna bacteria lah, yeah. So I think gambar dekat sini lah. So what type of strains that you use are here, yeah. So the test principle is here. So it's a very simple test also. So perhaps I can ask for you to get familiar with this, yeah. So ini salah satu representation also lah. how we can use AIMS test to actually look at genotoxicity punya effects. Okay, so here is also about the mouse lymphoma assay. Yeah, so another type of in vitro test. Yeah, so additional test is perhaps looking at the nucleus, yeah, micronucleus test. Yeah, and yang ini yang I cakap tadi. I was looking at DNA fragmentation. So I beli laboratory kit to yang nama dia memang COMET assay. So we were looking at how the DNA can get fragmented upon exposure to certain chemicals. Yeah, so it's very, very interesting actually. So here it is. Yeah, so you can see it. So maknanya the nucleus dari DNA gets fragmented. So fragments dia tertinggal kat belakang and memang nampak macam comet. Tahu kan comet? Okay.
Tahu.
So tahu ya. So that's why. So it's very interesting sebenarnya. But the kits, the lab kits too can be very expensive. Yeah. So nak test 50 sampel saja. I dah lah ada masa tu berapa groups of mice so yalah we cannot do like one mice, nanti result tak valid kan. We have to do like macam 3 mice and average kan average the data. So itu untuk test satu dose of the substance that I was testing lah. But I needed to test different doses. Yeah, so that is why doing in vitro ada pros and cons dia. Yeah, so sometimes it's also macam very expensive. Yeah, so you really have to like look at what you can do lah. Yeah, in terms of your research. Yeah. Okay, so other types of in vitro tests, perhaps looking at how the chromosome can be affected. Yeah. So you can read about the CHO punya testing. Yeah, the Chinese hamster ovary cells. Yeah, so what sort of cells are being used now to detect effects of mutations on the chromosome. So this is like some examples lah. Okay, so when you have chromosome aberrations, you get breaks, you get fragments. Yeah, you can get dicentrics. Yeah, okay, translocation. So another one will be the micronucleus assay. Yeah, so the nucleus will have its own nucleus yang kecil kat sebelah ke. So that is like what we can see as output from the micronucleus assays. So this is for you. Okay, so this is where I got the information from lah. So you can have a look on your own. Okay. So basically itu sajalah sebenarnya ya. So will you be okay in terms of talking about in vitro testing? Students? I think itu saja sebab the rest we will do tomorrow. Yeah, so we will look at health risk assessment and also yang inilah tomorrow. Okay, so I will stop sharing. Yeah, so it's nearly 11 or so. I do not intend to do class more than an hour sebenarnya. Yeah, but ada apa-apa ke you nak tanya saya about in vitro testing? Just now. Yes, Nurul Ain ke? You want to ask me anything? Yes,
about in vitro. Before this, kan ada assignment about toxicity testing juga, about untuk buat research on toxicity testing. So one of the vision is to to create like ada kaitan dengan in vitro testing juga, which saya ada baca another article questions about the cost of in vitro testing. Is it like is it true that it is more kurang cost daripada guna haiwan?
Itulah.
It need still kena ada lots more data and also kids and so on. So like kiranya replacement with in vitro testing tu adakah efficient or not? Tu je.
Betul, betul. I'm assuming that is a comment, yeah, not just not a specific question, kan, yeah? Because I think you got it right, lah, yeah. Yeah, very good. So meaning kan, it's not like macam a total replacement kan to in vivo testing kan, yeah. And also there are challenges, yeah, when you are doing a in vitro study, yeah. So the cost is one thing, yeah. So it's not like cheaper than when you are doing animal in vivo studies, yeah. So you can do check and balance lah. You have to look at especially the objectives of the research that you want to achieve, yeah. And then dekat situ lah the challenge-nya, yeah. As biomedical scientists, you really have to look at your objectives, and then you have to select your methods. Yeah. Now dah lah kan macam-macam approach ada, macam-macam methods ada, yeah. So you have to really think about, do the planning, yeah, so that in the end you will be able to achieve your research objectives, yeah. So there are pros and cons for both in vivo and in vitro lah. I think, as you know, yeah. Nurline, when you even read the paper that I gave you, the 21st century punya vision tu kan, ya. But mana tahu you all become biomedical scientists nanti yang akan buat kits-kits yang lebih murah ke, ya, yang give you rapid results ke, ya. So that is in terms of cost lah. Okay. Alright, so thank you. So any other issues ke? With regard to in vitro testing? Just now, I ada macam exposed to you jenis-jenis, ya. So perhaps you can take some time to look up on the experiments in the internet, ya. Apa dia AIDS test, apa dia yang apa tu lymphoma, mouse lymphoma assay, ya. So get yourself familiar lah with the type of in vitro punya testing that are around, yeah. So sit down and do some review, okay? Boleh kan, brothers and sisters?
Boleh.
Yeah. Okay, take time, ya. Macam familiarize sikit lah yourself, ya, dengan jenis-jenis in vitro testing ni. So depending on macam what is it that we want to achieve. So kalau nak buat genotoxic test, guna test apa? So macam tu, ya. Kalau nak buat cytotoxicity test, what sort of in vitro test we can use, ya. So that really needs for each student to actually be responsible for your own learning lah, ya. So there are a lot there, but what I give you as a guidance lah. Memang itu yang very famous and basic punya type of testing lah, okay. But again, I will need to emphasize, it all depends on what you are trying to achieve in your research. Sebab itulah your research objectives becomes very important. Perhaps the first thing that you put down also. Okay? Boleh ya? Students? Boleh faham ya apa saya cakap ni?
Boleh.
Boleh ya?
Boleh.
Very good.
Faham.
Okay. So this is like to prepare you lah ya. Bila you buat research ya, project tahun depan ke year 4?
Year 3.
Year 3 kan? Year 3 kan? Ya. So see, you have to be prepared lah, yeah, for this. So of course, start looking at what you would like to do, yeah. And of course, you have that option to go into diagnostics or research, yeah. So depending on what your interest would be, yeah. So start thinking about your future. Okay. All right. So anything else about in vitro testing? Yang tadi yang 3R replacement tu okay ke? Yes, Syami, apa dia?
Saya nak tanya macam kan sampai sekarang vaksin COVID-19 masih lagi macam tak digalakkan untuk ibu yang mengandung dan ibu yang menyusukan kan Prof. Sebab masih belum ada macam testing yang boleh membuktikan side effect dia tak teruk ke Prof. Jadi boleh pakai in vitro ke?
Boleh pakai in vitro? What do you mean?
Boleh buat maksudnya boleh testing dekat mice juga ke kalau macam mana macam nak tengok side effect tu.
But you know kan, COVID ni came on suddenly, and then the manufacturers of vaccine ni they had to actually come up with vaccines very quickly, yeah. But of course, kan when we read about how they come up with the vaccines, they did all the required testing lah, yeah. So I'm very sure they have tested in animal labs kan, yeah, because it's very crucial for safety and toxicity testing before we actually give this to humans kan, yeah. But of course time to do all that is very short, yeah. So mungkinlah tak tercapture effects yang macam selainnya. So that is why there is that concern on whether or not pregnant ladies are actually susceptible to long-term effects ke apa ke, ya. But we can only know very much later kan, ya. But of course vaksin yang mana yang katanya will not be advisable for mothers or breastfeeding mothers to take ke, macam mana, pregnant ladies ke, Syami?
Saya tak sure tu, saya baca dekat website.
Kan dia ada kan banyak-banyak jenis kan, ya. Kita ada yang Pfizer, AstraZeneca. So cara pembuatan vaksin tu pun different ya. Ada yang guna mRNA saja, ada yang guna the attenuated whole live virus, ya. So actually memang kalau research ni it needs time, ya. But when it's ad hoc like this, yeah, yes, possibly ada a certain percentage of humans receiving these vaccines will be affected by certain symptoms ke apa ke, yeah. So you can expect that lah, yeah. So keep on reading. It's a very good thing to do, yeah. So that you are aware lah, yeah, what is happening. So very important for us in this biomedical science field, yeah. So I hope it becomes like a motivation for you lah, ya. In this field, dalam program ni, you can contribute a lot to the ummah sebenarnya, ya. So I hope this will be like a motivation for you. Sebenarnya dalam time COVID pandemic ni, the biomedical scientists ni play a very crucial role, ya. How from not only being in the lab doing experiments ke, but how you can actually contribute to the community. Even now, I don't know, ada ke anti-vaxxers here ke? Yeah? So perlu ke sebenarnya people go for vaccines? Yeah, so we have that obligation there also, yeah, to do. So lagi anything anyone would like to share? Or any questions about in vitro? Boleh ya, nanti you do your self-learning kan, self-review. Semua sihat ke ni?
Boleh.
Everyone okay?
Sehat. Okay, Alhamdulillah.
Tak keluar rumah ya, I mean unnecessarily lah, ya, because of lockdown semua ni, ya. Don't unnecessarily get yourself exposed, ya. Memang kita as a whole nation has to come together lah to break this to flatline lah, ya. Reduce number of positive cases daily. So itu saya nak pesan lah, ya, pada all my students. But hopefully at the same time you are all healthy, ya. Sihat ya, tak demam ke apa ke. Dah vaksin ke? Ada yang dah dapat vaksin ke?
Belum, Prof.
Oh, okay.
Pantai Timur lambat.
Ya, yelah, tapi nanti mungkin UIA sendiri akan jadi macam pusat pemberian vaksin lah. So kan kalau you baca dalam news kan, KPT kan, Kementerian Pengajian Tinggi nak universiti macam kita ada SASMAC so we should be involved as a center to give vaccines pun so that is in the pipeline so I have also asked the Kuantan campus to look at this lah, sama ada kita punya own community, student, staff dapat dulu so kita tengoklah macam mana nanti so okay so kalau tak ada apa-apa, can I leave you at that and we jumpa tomorrow pula untuk the last session?
Boleh,
Yeah.
Prof.
You're all right, Okay. Okay, so thank you very much. We adjourn with Tasbih Kafarah and Surah Al-Nas. Okay, terima kasih semua. Thank you for your time. So I'll see you tomorrow. Pasti saya pukul berapa? 9:30 ke? But see you in our next session lah So Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Prof. Thank you, Doctor.
Thank you, Prof.
Thank you,
Okay, bye-bye.
Thank you, Prof.
Assalamualaikum. Take care. Okay.
Terima kasih, Doktor.
Welcome.